I'm Stanislaus Wong, and I'm a professor of chemistry at SUNY Stony Brook in Stony Brook, New York, with a joint appointment at Brookhaven National Laboratory in the Condensed Matter Physics and Materials Sciences Department. What you're going to be seeing in the next clip will be my presentation on the synthesis and applications of multifunctional nanomaterials, which was actually given at the European MRS meeting in Lille France in 2014. Anyways, we're going to talk about the synthesis and applications of multifunctional materials. And so the theme that I want to refer to is the idea of interfaces. So what I mean by interfaces is that there's interesting phenomena that tend to occur at boundaries. And what I'm going to talk about is hopefully strategies of designing a series of chemically linked nanoscale heterostructures using diverse and generalizable solution-based strategies, which is the point of this particular symposium. So it's the idea that synthesis can actually control structure property correlations, and the resulting oxide composite material that we fabricate often possess unique properties that are distinctive from and superior to those of the individual constituent components. And so what we're talking about is really a multifunctional architecture. So it's not really doping. It's not really a situation where we're creating a solid solution, but we're trying to maintain a structural differentiation between the two components which constitute our main heterostructure. So as examples, I'm going to first talk about a 1D silica nanotube 0D nanoparticle heterostructure. This is a multifunctional magnetic and optical construct which has potential for biolabeling. The second one is a tin oxide TiO2 fiber nano architecture, whereby the morphology can actually be tuned by variations in electrospinning conditions, and that has applicability in photocatalysis. And lastly, I'm going to end with a cerium phosphate quantum dot composite heterostructure, where these particular materials have potentially unique photoinduced charge transfer properties with the applicability for solar type of applications. Okay, so the first example is a multifunctional silica nanotube nanocrystal heterostructure. And essentially, why do I consider this particular material as a multifunctional architecture? Simply because that this construct has within it both quantum dots, cadmium selenide quantum dots, as well as magnetite nanoparticles, which are immobilized within a silica dioxide nanotube matrix. And superparamagnetic iron oxide nanoparticles have been used for protein harvesting, MRI, hyperthermia treatment, bioseparation, magnetic sensing. Quantum dots have been used for fluorescently labeled biolabeling imaging applications at the subcellular level. And all of these particular structures are actually incorporated within a silica nanotube matrix, which has prior and effective usage in bioseparations, drug delivery, and biocatalysis. And the issue with the silica nanotube is that it can be functionalized. Both the inside and outside can be functionalized to different degrees, and that's what makes this a very flexible construct. And this whole idea of this architecture of having a silica nanotube basically encrusted with quantum dots and iron oxide nanoparticles, well, this particular composite does have an aqueous stability of about 0.5 milligrams per mil in actual time periods of up to 1 day. So it's actually very useful for biological types of applications given this sort of stability. So just to give you a reminder what quantum dots basically are, the reason that quantum dots are interesting is that they have a size range of 2 to 50 nanometers, and essentially the properties of these particular quantum dots are dominated by their size. So what this means is that what you have is a shift to higher energy of the absorbance onset and the emission maximum towards smaller types of quantum dots. So what I'm showing you here are a series of cadmium selenide, indium phosphide, indium arsenide quantum dots, and as you can see, their absorption spectrum and emission spectrum basically vary as a function of size. And as you go towards smaller size nanocrystals, there's a shift of the emission maximum towards these higher energies over here. And so the optical emission of the cap nanoclusters, the arrows actually point to increasing size of these particular quantum dots. So what this means as a practical implication is that you could use quantum dots to label cells. As you can see, 2 nanometer size quantum dots are labeling the actual cell nuclei, whereas you have larger quantum dots basically labeling these actin fibers which protrude from the outside of these particular cell membranes. And so what we decided to do was to take this particular idea one step further. So we have a silicon nanotube matrix. The silicon nanotube is constructed by immersing an aluminum membrane in the presence of tetraethyl orthosilicate. In the presence of HCl and ethanol, you basically create your silicon nanotube, and then you immerse this in solutions of OTS and hexane, the OTS octadecyl trichlorosilane, in order to activate the wall of the nanotube so that it can incorporate the nanoparticles more easily. And then in subsequent steps, you're going to dip this particular silicon nanotube matrix in a solution of cadmium selenide in cyclohexane and also magnetite particles in cyclohexane in successive steps. And what this allows you to do is to incorporate various percentages, various concentrations of both quantum dots and iron oxide nanoparticles. And the one thing I should mention, these iron oxide particles and quantum dots have been pre-prepared. So I actually make these in previous steps. And what we see here is that these particular quantum dots have 3 to 4 nanometers in terms of diameter. The iron oxide nanoparticles are 12 to 15 nanometers in diameter, so that I can more easily differentiate and distinguish between these two types of particles. The second thing is that I'm also using different concentrations. 10 millimoles of quantum dots, 100 millimoles of iron oxide nanoparticles. And the reason I do this is to make sure that the silicon nanotube construct that I eventually obtain is going to be as biocompatible as possible. So that means that I minimize the amount of potentially deleterious quantum dot concentrations within that silicon nanotube composite. So once I do all these particular reactions, I have a silicon nanotube that has essentially varying concentrations of both iron oxide particles and quantum dots localized on their external surface. Okay? So what does this look like? And as you can see, the silicon nanotube and these composite heterostructures from the outside look more or less similar. From the SEM, scanning electron microscopy, these two composite structures look very similar, but I do have this one-dimensional motif of my silica dioxide platform. But when you look at the high-resolution TM, you can see that these composites basically have these particles sprinkled on the outside of the surfaces of these silicon nanotube walls. And what you see under these imaging conditions is that you have varying concentrations of these larger magnetite particles and these smaller quantum dots. And how do I know this? Because if I do elemental analysis, as you can see right here, the blue corresponds to these quantum dots. I mentioned to you that they were smaller, and these iron oxide particles are larger, 12 nanometers or so. These are 3 nanometers or so. These actual larger iron oxide particles, basically, when you can see elemental analysis, it has iron as you would expect. So we have the silicon nanotube base, and we have these varying concentrations of both iron oxide particles and quantum dots localized on their outside surfaces. Now, basically, the very cool thing about this is that I can make this quantitative. So I can vary by the loading densities, by the incubation concentrations, by the immersion times. I can vary the loading densities of both quantum dots and iron oxide nanoparticles directly on my silicon nanotube template. This is very important because basically nanoscience is just as quantitative as any other type of science, and what you want to do is you want to be able to control the relative concentrations of both so that you can actually make this overall construct as biocompatible as possible because I'm minimizing my quantum dot concentration. Now, what does this have in terms of implications for properties? Well, the first thing is that I can take a magnet and I can move this up and down, and I can move my composite up and down in aqueous solution. So that's actually one consequence, is that it is receptive to a magnetic field. The second thing is that if you actually looked at it by means of squid, what you basically find is that you have a signal which is very much similar to that of a magnetite nanoparticle, as you would expect. Okay, so a silicon nanotube by itself doesn't really have any sort of signal whatsoever. But basically, once you incorporate the magnetite particles, it does have a susceptibility which is corresponding to that of magnetite. The second thing is that you look at the emission properties of the quantum dots, and what you see is that with the silica, presence of silica, you basically have an emission peak, as you would expect, and also the silica oxide quantum dot magnetite heterostructure also has a signal, as you would expect, corresponding to the emission peak of cadmium selenide quantum dots. And silica dioxide with magnetite by itself basically shows nothing, again, as you would expect. So, in other words, that the presence of silica, presence of quantum dots and magnetite within the same heterostructure relates to the fact that it's not only magnetic, but it's also photoluminescent, and you could visualize this under an optical microscope, under scanning force, Anneker's scanning fluorescence microscopy, I should mention. Phase contrast image and the fluorescence image basically overlap. The control sample incorporating iron oxide alone basically doesn't actually show this particular signal. So as you would expect, the presence of quantum dots makes this visible. And the implication, therefore, is that you can incorporate these particular composites into HeLa cancer cells. You can image these HeLa cancer cells. And what I'm showing you right here is phase contrast as well as green and red fluorescence images of an individual HeLa cancer cell in the presence of nanotubes. And I know that my silicon nanotubes actually incorporate within these HeLa cells because basically, if I took a cross-section vertically, this is a set of vertical Z-stack images going from top to bottom, I'm actually shearing right through my cancer cell. I see a maximum amount of signal in slides 4 through 6, which basically means that most of my particular silica encapsulated nanotubes are incorporated within the interior of these particular HeLa cancer cells. So again, it's a very good bioimaging application type of construct. Okay, so the second example I want to mention is this idea of actually tin oxide TiO2. And this is actually fabricated using, this is a core shell nanoscale architecture produced by coaxial electrospinning. And in terms of conventional ways of making core shell materials, it often involves a two-step process whereby you make an initial core followed by a shell coating. And usually making core shell materials, you often have a tedious purification step, and it's often difficult to make a very good secondary homogeneous contiguous coating of a shell material. So what we basically did was we actually built our own coaxial nozzle for electrospinning, and the unique properties of our tubular tin oxide TiO2 system is that we have a well-defined heterojunction between TiO2 and tin oxide. We allow for efficient interparticle charge transfer, and essentially the hollow structure allows for accessibility of both the tin oxide and TiO2 to the reaction medium, thereby allowing for photo-generated electrons and holes to participate in the catalysis reaction. So this is actually a very different process. It's still solution-based in many respects, but we actually had to use electrospinning in order to really make a homogeneous core shell material. Just to remind you, electrospinning has actually been used to make products for filtration. It's been used to make scaffolds for tissue engineering, sensors and electrodes, batteries, solar cells, channels for channel tubules for drug delivery. And so what we did was, as I mentioned before, we actually created a novel core shell structure whereby we devised, we actually built this apparatus whereby we could insert into for the inner spinneret, for our core precursor material, tin ethyl hexanoate in the presence of mineral oil, and in the shell, we actually have titanium isopropoxide in the presence of PVP and ethanol. And so when we subject this to electrospinning conditions, we can actually devise a means whereby we can spin a core shell fiber whereby we have a tin oxide TiO2 construct where upon calcination we remove the polymer mineral oil. We have a metal oxide-based core shell material. This particular methodology is simple and versatile. It's efficient, it's relatively high throughput, and this potential process can occur under mild conditions. So we can actually spin as many fibers in as large quantities as we basically need and want. And the other cool advantage of using this particular process is that we can actually mix and match different energy levels of the core and shell material. In this particular case, our tin oxide and TiO2, we can actually mix and match the valence and conduction shell alignments so that we can create novel types of architectures again, as I mentioned earlier. So as we actually increase the concentration of tin, as I'm showing you here, what you see is that as you proceed from left to right, we're increasing tin precursor concentration. We actually, the morphology that we get of our electrospun fibers evolves from that of a hollow tubular coaxial structure to a pea-pod-like structure in the middle, and finally onwards to a solid filled core shell material, as you see in the final panel. And so what's again very interesting about this is that, first of all, we have this particular tin precursor, 1.6 millimoles per mil, and essentially when we do that, we have a core shell material whereby, again, the outside corresponds to your TiO2, the inside corresponds to tin oxide. You can see from the EELS plot that essentially what you have is more of titanium on the outside of the structure, the tin on the inside. The diameter of the entire fiber is about 300 nanometers. The inner diameter is about 138 nanometers. And what you could do is that you are actually able to spin fibers into mats, which have a hollow dual layer structure. They're highly uniform and flexible. And again, you can make these mats on a relatively macroscopic scale. So again, one of the advantages of this particular technique is that you can actually translate from a nanoscopic type of architecture all the way to something which is more macroscopic. Okay, so again, you increase the concentration of tin precursor solution to 2.2 millimoles per mil. And what you basically have here is a pea-pod-like material. And so the outer diameter of this is about 284 nanometers. The inner diameter is about 100 nanometers. And if you look at the EDS profile, you have, again, much more titanium on the outside. The inside is actually composed of tin. And I'm drawing you here the pea-pod-like motif. The tin oxide particles, which measure about 58 nanometers in diameter, are basically like these pea-pod-like structures located throughout the entire channel of TiO2. And this is, again, a very interesting motif when we increase the concentration of tin precursor solution to 2.2 millimoles per mil. And onward to 3 millimoles per mil, what you see with what you actually obtain is really almost like a solid filled core shell motif. The outer diameter is 275 nanometers. The inner diameter is 53 nanometers. And what you see is a distinctive boundary between the shell and the core along the entire length of that solid filled coaxial fiber. The outer surface is somewhat roughened, but the point of it is, again, you can see very clearly that you have a distinctive core surrounded by your porous outside shell. The outer surface is roughened. The arrows basically point to fiber cross-sections where the core and shell are concentrically wrapped. Okay, so again, if you take this EDS profile across that particular fiber, what you basically find is that you have the presence of tin in the inside, TiO2 on the outside. You do the mapping profile. What you find is that you have predominantly titanium on the outside, you have tin on the inside, a core structure. The core area is dominated by the tin signal, the outside is dominated by the titanium signal, but there is some degree of doping of either tin or titanium, either on the tin on the outside surface or the titanium signal with the inside structure. But the point of it is that these particular materials are polycrystalline, and essentially that we are creating a relatively uniform boundary between both the titanium and that actually tin structure. So the very interesting lesson to be learned from electrospinning is that you can actually create a variety of different morphological motifs simply by varying the conditions by which you do this particular electrospinning process. And one of the implications of that is that you can actually have a very high surface area material compared to, say, rhodium B, as you can see right here, when you're trying to photodegrade this particular dye. Our particular structures behave much better than, say, the conventional Degussa P25 nanoparticles. And it turns out the increase in reaction rate constant is due to the fact that what we have are higher surface areas relative to, say, some of the fibers or relative to, say, to some of the nanoparticles. So the increase in for instance, the porosity of our particular structures means that there's more active sites, more active area for the photodegradation process to basically take place. So that's one of the advantages, again, of basically able to tuning that morphology. Okay, so in terms of the last example I want to show, or before I get to that, I just want to summarize the TiO2 tin oxide fibers. They have a well-defined core shell structure which can be fabricated by coaxial electrospinning. Interior morphologies can be prepared by varying that precursor concentration, and the photodegradation measurements suggest that the tubular coaxial nanofibers are superior to those of analogs as a result of the formation of the heterojunction as well as the presence of the one-dimensional motif. The methodology is highly versatile in that it can be extended to these different binary metal oxide systems, metals or sulfides, and that these particular fibers can be directly electrospun onto these different substrates, thereby allowing them to be incorporated into various types of electronic fibers. So again, some of the key points to remember about this particular structure is that you can actually tune that heterostructure, so that heterojunction. So for instance, I chose TiO2 and tin oxide. I can actually choose other types of metal oxides, again, in order to basically mix and match these particular energy levels, which you see here, and that, moreover, they can actually be spun into fiber mats, which are going to be very useful for commercial types of applications, okay? So, in terms of the third example I want to highlight, it is the idea of looking at photoinduced charge transfer in luminescent quantum dot cerium phosphate metal oxide nanowire composite heterostructures, which we actually completed just this year. And to give you a background of why we think this is a particularly interesting type of architecture, I just want to highlight the fact that my group has a large effort in actually making these ternary metal oxide nanostructures. So this is a review from about 10 years ago, but basically we're interested in making these AXBYOZ nanostructures. So for instance, the principle is that we want to be able to control both morphology and composition simultaneously. So we want to make, for instance, bismuth ferrite, barium titanate, barium chromate. So we can actually make A is bismuth, A is barium, B is titanium, iron, any random set of either A or B types of elements we would want to incorporate into one particular composite structure, where we're able to control both morphology, chemical composition, and purity simultaneously. And I think that's actually quite useful and relevant. So one of the strategies that we're trying to employ, and many other groups do the same thing, so we're not unique in this, but we want to use relatively mild synthesis strategies in order to generate samples with high quality crystallinity and purity, with control over size, shape, morphology, as well as monodispersity as well. And so one of the ideas of this is the so-called green chemistry principle. So you want to use non-toxic precursors in aqueous solvents with either non-pyrophoric or stable precursors, relatively few numbers of reagents, minimization of reaction steps, high-yield processes with little of any byproducts under ambient temperature and pressure conditions with the possibility of scale-up. So one of the techniques that we developed in my laboratory was actually incorporating most of these particular steps, with the actually exception of scale-up. But the point of it is that we actually are able to generate nanowires under ambient conditions in aqueous solvents. And what we did was to develop a so-called U-tube method. So you can imagine that if, for instance, if I'm trying to make barium tungstate or barium chromate, what I would want to do is put an aqueous precursor solution of, for instance, barium chloride to the left, sodium tungstate, sodium chromate to the right. I allow these aqueous solutions to interact within the pores of my aluminum membrane, and what I end up getting, so the actual pore channels, the membrane channels of my alumina template membrane are the reaction vessels whereby the actual wires can form. And so what this means is that after I process my aluminum membrane, I can either generate an array of these particular oxide rods or isolated oxide rods. And to show you that this in fact actually does take place, this is a cross-section right through an aluminum membrane template, and what you basically see are actually these filled structures, these filled wires. So this particular process allows you to generate this monodispersed set of wires with little of any byproducts whatsoever. So I'm not getting any particles, I'm not getting any other type of motif apart from the wire that I'm trying to generate in the first place. And so what this means for us is that we actually applied the exact same thing with actually our terbium-doped cerium phosphate wires. We have in the one side our phosphate precursor. On the other side, we measure a stoichiometric amount of either cerium and the terbium dopant. In that particular case, we use a polycarbonate membrane, but we get the same sort of idea, that we get these particular phosphate nanowires in relatively high quantities. They're pure, they're monodisperse. I basically get the same wire as I'm getting in previous steps. And the very interesting thing, again, is that this happens under aqueous solution. So I'm able to get all these types of wires under aqueous solution conditions. Okay, so basically my wires are pure, they're single crystalline. I really have a very sharp diffraction pattern. My EDS spectrum shows me that I have terbium, I have a cerium and phosphorus, as you would expect. Copper comes from the TM grid, but as you can see, this is a morphologically pure, single crystalline type of wire that we're able to generate. So in terms of the actual structure of cerium phosphate, it actually stabilized by water molecules. I have a single phase of hexagonal cerium phosphate wires, and my quantum dots are basically localized on my external surface of my cerium phosphate nanowire platform. And the way that this particular cerium quantum dot immobilizes onto my cerium phosphate is actually by means of hydrogen bonding. And so just to prove that this, in fact, the quantum dots are localized on my cerium phosphate, EDS mapping shows that cerium, phosphorus, oxygen, cadmium, selenium are actually, all the signals are localized onto one particular architectural motif. They're all coincident. They all are coinciding onto one structure, as you can basically see here. And if you look at, again, at scanning fluorescence microscopy images, bright field images, and the fluorescence images, they're basically stacking on top of each other, showing you that you have a uniform distribution of quantum dots localized on the cerium phosphate platform. Okay? It's actually quite interesting in that respect. So IR shows us that the actual bonding takes place between the phosphorus PO bonds on topo, triacetylphosphine oxide, capping on my quantum dots. So it's the ligand-capped quantum dots, and it's actually linking with the intercalated water molecules of my cerium phosphate. So as I mentioned before, cerium phosphate actually had these water molecules which are dispersed throughout the structure. So these water molecules are bonding onto my particular quantum dots, and so as I can see, the shifts of OH and PO stretching modes in my heterostructures are consistent with that formation of my heterostructure involving both quantum dots and cerium phosphate. But the main point of why I want to show you this is that this structural formation has, again, implication for the properties of the overall material. So what I mean by this is that when I look at my quantum dot absorption profile, which is under illumination of 375 nanometers, whether I'm looking at my quantum dot or my terbium, and also my phosphate, cerium, and also my terbium type of emissions, all of these emissions basically go down. They actually all quench upon formation of my heterostructure. So whether I'm looking at cadmium selenide, whether I'm looking at D2F emission bands of my terbium or also my cerium, all of these emission bands basically diminish in intensity. So there's an efficient absorption of both cerium phosphate and quantum dots, but upon formation of my heterostructure, all of these particular bands actually will quench, diminish, attenuate in terms of intensity. And I can look at my PL quenching, I can look at my lifetimes in more detail by actually varying the amount of cerium phosphate which I have. And what I'm showing you here is that I'm showing increased PL quenching, shortened lifetimes with exposure to higher amounts of cerium phosphate. So these images are a little bit, perhaps a lot of data on this, but what you see as you go from this top signal, profile all the way to the bottom is that I'm increasing the amount of cerium phosphate which I have. So what this means is that I have more area, surface area of cerium phosphate to interact with my cadmium selenide quantum dot. And so what this means is that there's more of a possibility, more of a potential for the quantum dot photoluminescence to basically be quenched in the presence of cerium phosphate. It's exactly what happens. As I increase cerium phosphate, I see a greater quenching of my quantum dot, and moreover, my lifetimes go from 23.2 nanoseconds all the way to 22, 16.9, and 7.9 as I increase my amount of cerium phosphate to quantum dot ratio from 0.33, 0.66, all the way finally to 1. So again, as I increase my cerium phosphate concentration, relative to that of quantum dots. I get more and more quenching. My lifetimes decrease with all the way from 22 nanoseconds all the way to about 8 nanoseconds. Okay, so again, if you can see this happens whether I'm illuminating at 280 or whether I'm illuminating at 375 nanometers. So how do I explain this? I explain this because basically my valence band of my quantum dots, which are right here below, they're below the valence band of my cerium phosphate. And this is an ideal band alignment for hole transfer to take place from quantum dots to cerium phosphate. So under visible light illumination, 375 nanometers illumination, which I'm showing you right here, under that type of illumination conditions, I basically have hole transfer taking place from my quantum dot to my cerium phosphate. Whereas if I look at 280 nanometer excitation, which is right here, I'm basically accessing a situation because of the fact that the conduction band of cerium phosphate lies above the conduction band of quantum dots. In that particular case, the reduction in the emission of both cerium and terbium emission is via electron transfer going from the cerium phosphate to the quantum dot. So the very interesting thing I think about this particular heterostructure is that I can access both hole transfer or I can favor electron transfer depending on the nature of the illumination through which I irradiate my heterostructure. So under 280 nanometers, I'm actually going for hole transfer. Under 375 nanometers illumination, I'm actually going for more of this electron transfer. So 280 is electron transfer, and 375 is more of the hole transfer. So again, it just depends on how I align my energy band levels relative to the quantum dot and also the oxide that I'm playing with. Okay, so I'm just going to end because I'm running out of time. That basically our group actually makes a lot of different types of oxide materials, which is the focus of this particular symposium. We actually do a lot by means of molten salt template directed synthesis and hydrothermal synthesis, making a wide variety of oxide materials. A lot of them are actually binary oxides or perovskites. They're actually either commercially relevant or they're going to be relevant for a number of other types of applications. And the reason that we choose these methods is that these methods tend to be relatively simple, flexible. We're able to, they're generalizable. We're able to make large scales or arrays of these particular materials. And in that particular process, we make what I would call a toolkit of non-carbonaceous structures. And what I'm showing you here in green actually corresponds to so-called other examples of multifunctional types of architectures that we've made in the lab over the years. Anywhere from, again, magnetic and photoluminescent materials to materials which are actually very useful for fuel cell types of applications. And again, you know, we're employing these different types of strategies. And with that, I want to thank my students who were involved in all of the projects. In terms of the silicate work, it was at Hongjin Zhu, electrospinning Xiaowei Peng, and most recently the work I showed you with the cerium phosphate quantum dots was actually done by J.K. Han. I want to thank my collaborators and also folks who helped us with technical support. I'm supported by NSF in the past, and right now predominantly by the DOE Office of Basic Energy Sciences, and I want to thank you for all of your attention. Thank you very much.